Fisiologia, né, em celebração aos 75 anos do Instituto. E eu só vou passar algumas mensagens rápidas para os alunos da pós-graduação, porque está chegando o período de lançamento de notas, e como esse ciclo de conferências, na verdade, está relacionado, associado a uma disciplina, né, na pós-graduação, os alunos que já completaram as 15 conferências necessárias para obter o crédito, eles têm que, na verdade, mandar até o final dessa semana já, por e-mail, né, para o nosso e-mail cac@biof.fj.br, aquele formulário que a gente envia sempre junto com a programação, preenchido e informando quais foram as conferências que vocês assistiram, né, e preenchendo as outras informações do formulário. Então, tem que fazer isso até o final da semana, se quiser que o crédito seja computado nesse período. Mas, para quem não completou ainda as 15 conferências, não tem problema também, porque, na verdade, essas conferências vão acumulando ao longo dos semestres, e no momento que completar as 15 conferências, é só, então, novamente preencher esse formulário, enviar para a gente, e a nota será computada. Mas, se quiser que o crédito seja computado já nesse período, tem que enviar, então, para a gente o formulário preenchido até o final dessa semana. Então, acho que essa é a única... acho que era o único aviso que eu tinha para dar, acho que não tem mais nenhum. Acho que no final da conferência passada eu já fiz essa apresentação, mas acho que já tinham poucas pessoas aqui, então gostaria novamente de apresentar o novo membro da CAC também, né, o professor Pedro Torres, que iniciou, né, na semana passada com a gente e está fazendo parte, abriu ali o vídeo. Tudo bom, Pedro? Então, nós somos agora 4, né, junto com a Daniela e com a Miriam. Bom, então acho que era isso, e vou então iniciar a apresentação do Dr. D. So it's a great pleasure, a great honor to have Dr. Dale Abel here with us. Thank you again, Dr. Dale. Dr. Dale is the chair of the Department of Internal Medicine and the director of the Fraternal Order of Eagles Diabetes Research Center at the Carver College of Medicine of the University of Iowa. At the same university, he holds the John B. Stokes III Chair in Diabetes Research and the François M Chair in Internal Medicine. So Dr. Abel has an MBBS with distinction from the University of West Indies and also a PhD in Physiology from Oxford University. He also completed internal medicine residence and chief residence at Northwestern University and also endocrinology fellowship at Harvard Medical School. So prior to moving to the University of Iowa in 2013, Dr. Abel was chief of the Division of Endocrinology and Metabolism at the University of Utah, where he held the H.A. and Edna Benning Presidential Endowed Chair in Medicine. Dr. Abel has been the recipient of numerous awards. Here I'll just highlight a few of them, such as the Established Investigator Award from the American Heart Association, Distinguished Mentor Awards from the University of Utah, the University of Iowa, and also the Gerald Aurbach Award for Research Excellence from the Endocrine Society. And also he is the immediate past president of this society. He also a member of the National Academy of Medicine, and also he was appointed to the Advisory Council of the National Heart, Lung and Blood Institute in 2016. His research program is focused on the cardiovascular complications of obesity, and he has been continually funded by the NIH for more than 20 years. So it's really a great pleasure to have you here, Dr. Abel. And before we continue, I'll just invite our director to say a few words. Thank you again.
Okay, Pedro. Thank you. Good afternoon. Welcome everybody here at the audience. Dr. Abel, it's a great honor for our institute to have your presence in the celebration of the 75th birthday. In the name of our director, Professor Bruno Dias, and all our academic community, I would like to thank you very much for the opportunity to see such an actual and exciting topic. I would like to call Dr. Carmen Pazos Moura, who will make extra information about your introduction. So please, Dr. Carmen, can you talk some words in order to introduce Dr. Abel? Thank you, Dr. Abel. I hope you all enjoy the conference here at the Google Meet, as well as at the YouTube channel. So let's go.
So, Dr. Dale Abel, allow me to call you Dale. And I can tell you how much pleased I am to have you here today. It's very special for me, really. And actually, it's the second time Dale lectured for us at the Institute. I think you remember you were, I think the first time was 1999 or 2000. I can't remember exactly. But, well, I first met Dale more than 20 Boston during his brief passage through the thyroid field. Well, he was a brilliant young scientist, actually one of the brilliant minds I ever met. At that time, he was involved in creating transgenic animal models, which was the cutting-edge technology at that time. And so he was an expert in making all these constructs to generate the transgenic animals to address relevant questions of in vivo action of the hormones such as insulin and thyroid hormones. And I take the opportunity to thank you, Dale, for all you taught me and tell you how much important it was for my subsequent work in my lab and the mentorship of my students. So Dale's pioneer's work resulted in outstanding contribution right in the beginning of his career, and his first author of high-impact papers published in Nature, Circulation, GCI, Diabetes, and so on. So it was very easy for us to foresee at that time the very successful career he actually had since then. Yes, he for the years, he focused in his studies in understanding the metabolic alterations in the cardiovascular system in diabetes, insulin resistance, and obesity. And his outstanding contributions are reflected in thousands of citations, many awards, and invitation to talks all over the world. And he's certainly a very well recognized leader in the field. And also, Dale actually is has a strong leadership, has an honest, firm character, and he's not afraid to speak his mind. And I believe that that led him to this characteristic that led him to the top of the representative position in endocrinology in the United States, which is the presidency of the Endocrine Society. So Dale, I'm very happy you are here. I'm sure everybody will enjoy your talk, and with no more delay, we ask Dale to start and your conference that we are so eager to hear. Thank you again.
Obrigado, Carmen. Thank you so much. Bom dia, everybody. It's a real pleasure to be with you, albeit over Google this morning, to really participate in this very important celebration of your institute. Let me start by really congratulating you for 75 years of leadership in science in Brazil, and really wish you all the best for the next 75 years. What I thought I would speak to you about this morning is some recent work and interest that we have been pursuing in our lab, looking at some novel mitochondrial mechanisms for cardiometabolic disease. So my lab has been very interested for many years in diabetic complications. And for those of you who study diabetes or think about diabetes, most of the suffering from diabetes comes from its complications. And for many years, the field focused heavily on the microvascular complications, such as diabetic retinopathy that leads to blindness, diabetic nephropathy that leads to end-stage renal disease, diabetic neuropathy that leads to traumatic amputations and pain. But really what drives the mortality in individuals with diabetes is the increased risk in macrovascular complications, classically studied as the increased risk of cardiovascular events such as myocardial infarctions and strokes. It turns out that after myocardial infarction, the second most prevalent macrovascular complication of diabetes, in fact, is heart failure. And so heart failure is now increasingly being recognized as a diabetic cardiovascular complication. Let me show you one of many examples to illustrate this point. So this is a study that was published recently out of Scotland. And it looked at individuals across the entire population who presented to the hospital with heart failure. And the blue line shows individuals who are not diabetic. And this particular slide is showing individuals who had no prior history of ischemic heart disease. So you can see clearly heart failure increases with age in everybody. But if you are diabetic, as shown here by the green line for type 1 or the red line for type 2, there's a significant increase in the risk of heart failure if you have diabetes. And that increase in risk starts quite early, even in the 40s and 50s, and then it really amplifies as individuals age. So we have for a long time been very interested in understanding the pathophysiology to make the point that multiple pathways contribute to the increased risk of heart failure in diabetes, as shown here. It's almost as if the heart is dying by 1,000 flogs, and almost every significant pathway, as shown on this slide, has been demonstrated by many groups to contribute to the risk of heart failure and diabetes. Our group has really focused over the years on these 4 areas: alterations in insulin signaling, mitochondrial dysfunction, lipotoxicity, and increased fatty acid utilization as potential causes or contributing factors to the increased risk of heart failure in diabetes. And to summarize many, many years of work with regards to cardiac metabolism, that what we and many others have shown is that in diabetes, there's a switch in myocardial substrate use that increases the dependence of the heart on fatty acids. And so there's increased fatty acid uptake, increased delivery of reducing equivalents after beta oxidation to the mitochondria. However, we and others have also shown that there is mitochondrial dysfunction that develops in the context of diabetes, characterized by impairment in oxidative phosphorylation that then leads to overproduction of reactive oxygen species that leads to multiple impacts within the heart, such as not only oxidative mediated damage of proteins and lipids, but also functionally leads to uncoupling of the mitochondria, leading to increased oxygen consumption rates, which again increases the vulnerability of the heart to injury in the context of ischemia. So more recently, we have been focusing on some other aspects of mitochondrial biology, and I'll start the talk by talking a bit about some work that we've been doing in the area of mitochondrial dynamics. And so what is mitochondrial dynamics? So as many of you are aware, mitochondria are not these kind of static organelles in cells, but they're very dynamic in the sense that they undergo repeated cycles of fusion and fission. And these cycles are modulated by distinct proteins that mediate these signaling pathways. Specifically, DRP1 is a cytosolic protein that translocates to the mitochondria. There's a receptor called FIS1, and this interaction leads to mitochondrial fission, whereas mitochondria can also undergo fusion. And there are outer membrane GTPases known as mitofusins that mediate fusion of the outer mitochondrial membrane. And then OPA1 or optic atrophy 1 mediates fusion of the inner mitochondrial membrane. OPA1 also plays a very important role in the maintenance of mitochondrial crystal integrity. So if you look at this electron micrograph, which is taken from hearts of a diabetic mouse model, specifically the DB mouse, which, as you know, has a mutation in the leptin receptor. And these animals become very hyperphagic, very insulin resistant, and developed severe type 2 diabetes. You will see that the mitochondrial morphology is really quite different compared to the wild type animals shown here on the left. So you'll see that the pattern of the mitochondrial network has changed. You'll see that the density of the cristae have changed, and you'll also notice that the mitochondria are just sort of irregular in their overall appearance. I'm just going to change to my laser pointer here. Right. Okay. So this is a characteristic of what we see in the heart in this diabetic model. Now you'll also notice that there are differences in mitochondrial size here, and I'm going to come back to that because this could potentially be could imply that perhaps the way that mitochondria are fusing and dividing could be perturbed. So we recently completed a study where we were very interested in understanding the contribution of mitochondrial dynamics in the heart. And this was work that was done by Kensuke Tsushima when he was a postdoc in the lab. And Kensuke is now an assistant professor at the University of Tokyo. So to address this question, we looked at an animal model of lipotoxicity. You recall that I mentioned that diabetes is characterized by increased lipid uptake into the heart. And so we collaborated with Jean Schaefer, who was at WASH at the time, and studied an animal she had generated that overexpressed this enzyme, acyl-CoA synthase. When fatty acids enter a cell, they are conjugated to acetyl-CoA to make fatty acyl-CoAs, and the enzyme that mediates this is acyl-CoA synthase. And you can see here on this PET scan in the mouse that there was an increased uptake of fatty acids into the hearts of these animals compared to the control animals. And that was about a twofold increase in palmitate uptake, which is about what we see in the context of diabetes. Interestingly, there was no increase in triglyceride content, but a significant increase in the content of ceramide and the content of diacylglycerol in these hearts. One of the things that we noticed in these animals was that when we looked at their mitochondrial diameter or mitochondrial size, when I show you the pictures in a second, they appeared to be locked in a fetal state. So if you look at the mitochondria in wild-type animals from the time of birth throughout weaning and then into adulthood, there is a significant increase, almost a doubling of mitochondrial diameter by the time these animals become adolescents, and they maintain that. But these animals that were lipid overloaded, and again, the promoter here was the myosin heavy chain that drove the expression of acyl-CoA synthase. So it was turned on from embryonic day 12. You can see that the mitochondria appeared to be locked in the small size state. And we also saw, however, that there appeared to be more of these mitochondria as evidenced by mitochondrial volume density. So here is the image. So this is the appearance of the wild type heart. Again, you see nice mitochondria, nice cristae that line up parallel to the sarcomeres. But in these acyl-CoA synthase transgenic animals, you can see that these mitochondria really are small. And in fact, it almost gives us a general appearance that the mitochondrial network is fragmented. And so when we saw this, we wondered whether or not this was a manifestation of altered mitochondrial dynamics. And specifically, could this be a manifestation of either increased mitochondrial fission or decreased mitochondrial fusion, or perhaps a combination of both things. And again, I want to remind you of the key players in this process. OPA1 for inner membrane fusion, mitofusins or MFNs for outer membrane fusion, and DRP1 and FIS1 for mitochondrial fission. One other important point to bring to your attention at this point is that DRP1, which normally is cytosolic and shuttles to the outer membrane of the mitochondria to promote mitochondrial fission, is also subject to post-translational modifications, including phosphorylation. And I show you one phosphorylation site here, which is DRP1 at serine 637, which is mediated by protein kinase A. which actually is anchored to the mitochondria by a protein called a couple of years ago now. What we found was that the lipid overload in these hearts led to a few things. First of all, it led to an overproduction of mitochondrial reactive oxygen species, and these mitochondrial reactive oxygen species led to altered processing of OPA1, and also led to proteasomal degradation of ACAP121, which therefore reduced the phosphorylation of DRP1 and serine 637, meaning that DRP1 was constitutively on the mitochondrial membrane, leading to mitochondrial fragmentation. Now, when we initially had written this paper, the reviewers were a little bit skeptical about mitochondrial fragmentation and forced us to more rigorously define what that meant in the heart, and I will show you that in the next the next couple of slides. But the other reason why we believe that ROS was an important mediator of this phenomenon in terms of mitochondrial size regulation was that we also looked at a mouse model that overexpressed the antioxidant superoxide dismutase, or SOD2. And if you look at this very carefully, what you see here is that in the SOD2 transgenic mice, the mitochondrial diameter is actually significantly larger than in the control animals. And this is quantified over here as mitochondrial size. Whereas in these acyl-CoA synthase transgenic mice, as I mentioned before, the mitochondrial size is significantly smaller. We then crossed the SOD2 transgenic mice with the acyl-CoA synthase transgenic mice, and in fact what we saw was that we normalized the mitochondrial diameter as shown here. Of course, the other thing is that, you know, as mitochondria get bigger, there are fewer mitochondrial structures. So in the SOD2 transgenic animals, the mitochondrial number appeared to be lower, whereas in the acyl-CoA synthase animals, the mitochondrial number appears to be higher. And of course, in the cross of those two animals, we maintain or restore the normal number of mitochondria. Now, as I mentioned before, the reviewers were skeptical in terms of, was this really mitochondrial fragmentation? And so they forced us to really take a harder look. And so what we ended up doing was we collaborated with a group in Germany that does three-dimensional electron microscopic tomography. And so it's like putting a virtual headset on and looking at the mitochondria in multiple dimensions. And this was actually a very informative experiment, because what we learned was that on the left here is the normal mitochondrial network. On the right is what happens in these acyl-CoA synthase transgenic mice. And if you look closely at the mitochondrial network, what in fact you see is that these mitochondria are thinner, but they are prolonged and really almost look like spaghetti or noodles. And in fact, let me give you a bigger picture of that, that when you have the lipid overload, where we have activation of these mitochondrial fission pathways, what we in fact see in the heart, which doesn't have a lot of space for mitochondria to bud off and go everywhere in the cell, is we see that there's a stretching and a narrowing of the mitochondrial network. And so it then goes in and out of the field. So when we do a two-dimensional section, what we see is what appears to be multiple small mitochondrial fragments, but in 3 dimensions, in fact, it's really a reorganization of the mitochondrial network that really shows mitochondria thinning out and becoming increasingly tortuous. So this is what mitochondrial fission actually looks like in the intact heart. And so what this study taught us was that fatty acid-derived reactive oxygen species drives signaling pathways that modulate mitochondrial form and bioenergetic function metabolic homeostasis. And you know, it gives me tremendous pleasure to share this work, which was spearheaded by Renata Pereira, a proud PhD graduate from the Federal University of Rio de Janeiro, and who worked with me as a postdoc in my lab for many years and is now an independent faculty member, just received her first NIH grant. So what Renata studied was the impact of altering the expression of OPA1, which in the heart, in skeletal muscle. And the rationale for that was because, as many of you know, that mitochondrial dysfunction is a characteristic of insulin resistance. The muscle, we would get insulin resistance. Well, we actually were wrong. What in fact we saw was that in these animals where we reduced OPA1 expression, not unexpectedly, we saw a disruption of mitochondrial cristae, as shown here. But when we put these animals on a high-fat diet, it turns out that the animals that were deficient in OPA1 in skeletal muscle did not gain weight, whereas the wild-type animals that you can see here gained significant amount of weight. So the question then is, what was happening in these animals that prevented them from weight gain? And when we looked in metabolic chambers, what we saw was that these animals had a significant increase in energy expenditure. Moreover, these animals are also protected against diet-induced diabetes. This is a glucose tolerance test curve. You can see that the wild-type animals on a high-fat diet are significantly hyperglycemic, whereas the knockout animals shown here in red on a high-fat diet are not hyperglycemic and so are protected from obesity-induced diabetes. Similarly, hyperinsulinemia is a hallmark of insulin resistance, and this is a characteristic of that were deficient for OPA1 in their skeletal muscle. So we then wondered, mechanistically, what was the link between altering the expression of OPA1 in skeletal muscle and this metabolic protection that these animals were manifesting? And so we looked at many things, and we landed on a novel protein called FGF21, or fibroblast growth factor 21. And we looked first at the messenger RNA of FGF21 in skeletal muscle, and we saw that there was a significant increase in FGF21 mRNAs, as you can see here in the knockouts, whether or not they were on a controlled diet or on a high-fat diet. Now, one thing to bear in mind is that skeletal muscle ordinarily expresses very, very little FGF21. So any increase in FGF21 that one sees, it could be meaningful, it could not be meaningful. And so we went on to measure circulating levels of FGF21. And you can see that there was a significant increase in circulating levels of FGF21 in the knockout animals that persisted when the animals were on a high-fat diet. Now, under normal physiologic circumstances, FGF21 is usually made in the liver, and so there was some skepticism as to whether or not this was really coming from skeletal muscle. And therefore, we looked at the mRNA expression of FGF21 in the liver. You can see that in wild-type mice with high-fat feeding, there's an increase in FGF21. In knockout animals, the liver FGF21 expression is about the same as one sees in high-fat fed wild-type animals. So there's no induction of FGF21 in the liver. And the other source of FGF21 is white adipose tissue. And again, you can see there is no increase in FGF21 in white adipose tissue relative to wild-type mice on a control or high-fat diet. So what is FGF21? So FGF21, or fibroblast growth factor 21, is a hormone that has multiple pleiotropic metabolic effects. It has profound effects on the liver to increase liver fat metabolism. It has effects on adipose tissue, specifically to promote the conversion of white adipose tissue to beige or brown adipose tissue. And all of these changes lead to increased energy expenditure. It has effects on bone and beta cells. It has a very important effect on the brain to regulate food intake and specifically to regulate appetite for sweets. So the question then is, everything I showed you so far, and the reviewers were arguing with us on this, was that we see more FGF. This increase in the circulation come from skeletal muscle. And so to prove that, we generated another mouse model where we inducibly deleted FGF21 and OPA1 in skeletal muscle together. And so you can see here when this was successfully achieved that here are the OPA1 knockout mice that have increased levels of FGF21 in muscle, whereas in these double knockout animals, we prevent this increase in FGF21 in muscle. That's by Western blot, and of course in the circulation we have a reduction in FGF21 levels. So that really proved to us that this was coming from skeletal muscle. And you can see then when we put these double knockout animals on a high-fat diet, their FGF21 levels were really the same as wild-type animals on a high-fat diet. And importantly, this protection against obesity, which I showed you before in the OPA1 knockouts, was completely prevented in these double knockouts. You can see here that they get as much weight as wild-type animals on a high-fat diet, and they become as diabetic animals on a high-fat diet as shown here, and they become as hyperinsulinemic as wild-type animals on a high-fat diet as shown here. So the question then is, mechanistically, how are we linking OPA1 with this increase? When we knocked out OPA1 in muscle, we got activation of the endoplasmic reticulum stress, or the unfolded protein response pathway. And this is one manifestation of the increased ER stress pathway. This is a BIP, which is a marker of increased ER stress. And so we said, well, what if we prevented ER stress? And we did this by treating the animals with a chemical chaperone called TUDCA, and this normalized ER stress. But in parallel with that, it also reduced the protein levels of FGF21 in the skeletal muscle and normalized serum levels of FGF21. So the model then that we have here, and again, here's a picture of Renata, is that mitochondrial stress induces mitochondrial dysfunction, leading to ER stress. And we now have very good evidence that this ER stress activates ATF4 that binds to the FGF21 promoter, leading to an increased secretion of FGF21, leading to diet-induced resistance, to resistance to diet-induced obesity and insulin resistance. So it turns out that FGF21 is in fact now a clinical biomarker for many mitochondrial myopathies. And this is work that was spearheaded in Finland by Anna Suomalainen. And this is one example of a recent publication from her group in Cell Metabolism, where they looked both at patients as well as mice with mitochondrial myopathy. And what they saw was that there was this integrated stress response that led to a transcriptional response that induced the secretion of myokines such as FGF21 and GDF15, and that FGF21 in particular, they showed had profound effects to alter intermediary metabolism, also had effects on leading to browning of white fat, the regulation of lipolysis in the liver, and the increasing glucose uptake into muscle, heart, and brain, as well as white fat. So this really, defines a novel hormonal pathway linking mitochondrial stress with metabolic regulation. Now, Renata has gone on to study OPA1 in other tissues, and I'm going to share a couple of data from her group now looking at OPA1 in brown adipose tissue. And so we had seen that if you put animals in the cold, that leads to induction of brown adipose tissue function. There's actually increased cleavage of OPA1. So we believe then that OPA1 clearly had a very important role to play in brown adipose tissue thermogenesis. And so to get a deeper sense of that, Renata generated animals with brown adipose tissue specific knockout of OPA1. You can see that here. And you can see that this is preserved in the white fat. And unexpectedly, if you look now at the knockout brown fat, so this is from the knockout animal, this is the normal brown fat. Normal brown fat looks like liver. It's very full of mitochondria. It's red, it's beefy, and you can see that the brown adipose tissue from the brown adipose knockout animals looked much more pale. And ultrastructurally, you can see that the knockout brown fat had a lot of lipid droplets relative to the mitochondria-rich brown fat seen in the wild types. And again, you can see these beautiful mitochondria in the wild types and these very unhealthy-looking mitochondria in the knockout animals. Moreover, she also observed that there is a repression of many of the transcriptional markers of thermogenesis, such as uncoupling protein 1, PRDM16, and type 2 deiodinase, but there's a compensatory activation of PGC1 alpha, and I suspect it's really an attempt to increase mitochondrial diagenesis in the face of defective mitochondria. And also, similar to what we saw in skeletal muscle, a significant induction in FGF21. So despite the fact that there was clear impairment in brown adipose tissue bioenergetics, as shown here, these are mitochondrial respirations from brown adipose tissue mitochondria, and you can see that the oxygen consumption rates in response to multiple substrates, for example, palmitoyl carnitine or pyruvate malate, was significantly reduced in these mitochondria. A wild-type animal, and you put it at 4 degrees, their body temperature goes down, as shown here. But these OPA1 brown adipose tissue knockout animals seem to be able to maintain their core body temperature. And in fact, when you put these animals on a high-fat diet, they also did not become obese. So you can see that wild-type animals gain weight, they accrue fat mass, whereas these brown adipose tissue knockouts did not accrue fat mass and did not gain weight. So despite the fact that the brown fat is dysfunctional, these animals appear to be behaving as if their brown fat is indeed functional. So the question then was, why was that happening? And it turns out that the increased energy expenditure that we observed in these animals that were lacking OPA1 in brown adipose tissue, and this is shown here as the energy expenditure both on a controlled diet or on a high-fat diet, had induction of browning in their white fat. So you can see now, we're looking now at white adipose tissue, and you can see that in the knockout animals, there is increased browning as evidenced by induction of uncoupling protein 1, protein and mRNA, induction of thermogenic pathways such as PRDM16 and type 2 deiodinase. And if you look here at the immunohistochemistry, you can see that the mitochondrial staining in the knockouts is significantly greater than the mitochondrial staining in the wild types. And this occurs in parallel, of course, with induction of the gene pathways that would promote mitochondrial biogenesis and fat oxidation, such as PTC1 alpha, CPT1 beta, and PPAR alpha. So what this study really taught us then was that in brown adipose tissue, mitochondrial stress also induces the secretion of FGF21, which is therefore a batokine. And in addition to FGF21, induces the secretion of other batokines such as GDF15. And in fact, this leads to the browning of white adipose tissue that promotes this increase in basal metabolic rates and maintenance of glucose homeostasis, and of course, a resistance to diet-induced obesity. So it really underscores really an emerging biology linking mitochondrial dynamics with metabolic regulation in mitochondria-rich tissues such as brown adipose tissue, and as I showed you earlier on in skeletal muscle as well. For the last bit of my talk, I want to switch gears a little bit and talk about mitochondria and their adaptation in heart failure. And they transition from physiological cardiac hypertrophy to pathological cardiac hypertrophy to heart failure is really characterized by profound changes in mitochondria, so that whereas in physiologic hypertrophy, such as exercise, there's an increase in mitochondrial capacity driven in part by PGC1 alpha, PI3 kinase, and AMPK, that in pathological cardiac hypertrophy, there is a clear evidence by us and many other groups of decreased mitochondrial oxygen consumption. And certainly by the time you get to heart failure, there is a profound collapse of multiple regulatory pathways that drive mitochondrial metabolism. And it is believed that this might contribute to the reduction in energy supply that may in fact contribute to heart failure. So the widely held notion then was that because the heart, being a contractile pump, requires multiple times its weight in ATP per day to maintain contractile function, that in the failing heart, that the ATP deficiency might in fact drive contractile dysfunction. Now that is true to an extent, but we argued actually quite a few years ago that when you reduce mitochondrial capacity, you also get spillover of metabolites into other pathways. Shown here, for example, in this cartoon, that the pentose phosphate pathway, the hexosamine biosynthetic pathway, that could also secondarily impact myocardial contractile function. So to begin to get a handle on this, we conducted a series of experiments looking at metabolic profiling in failing hearts. And this was a paper I was involved in with colleagues at the University of Utah, where we took advantage of the fact that when patients with heart failure are having a left ventricular assist device or LVAD device implanted, we can get a biopsy of cardiac muscle. And in this study, what was done was that a biopsy was done at the time of LVAD implantation. And for those patients who either went to transplant or recovered, we then took another biopsy at the time when the LVAD was removed. And very interestingly, we saw a unique pattern of metabolism in these hearts. Specifically, we saw that there was an accumulation of pyruvate, an accumulation of what appeared to be glycolytic intermediates, but a reduction in citrate, which is a marker of TCA activity and function. And in fact, when these hearts were subsequently unloaded and we got the second biopsy sample, we actually saw a further exacerbation of this metabolic mismatch. More recently, we have also looked at another independent group of failing hearts. So these were hearts from the University of Pennsylvania, where we took donor hearts from their heart transplant program, and these hearts were ultimately not used, and then explanted failing hearts that we had access to. And you can see some of the clinical characteristics of these patients here. But again, shown here is that these hearts, these are patients with heart failure with reduced ejection fraction. You can see that there's a significant reduction in ejection fraction and increase in cardiac mass. So we did metabolic profiling on these hearts. This is a volcano plot showing the metabolomics experimental results. But what this arrow shifted slightly, but basically we saw that there's an accumulation of sugar-derived or glucose-derived metabolites such as sorbitol, mannose, galactonic acid, and galactose and galactosamine. So it appeared as if there was this accumulation of these glucose-derived metabolites in these failing hearts. So let me just show how what that looked like when we quantified it. So we saw that there was an increase of free glucose, an increase in glycerol 3-phosphate, a reduction in lactate, and a reduction in TCA cycle intermediates such as fumarate, malate, succinate, for example, and a reduction in alanine. And moreover, we saw that metabolites downstream of glucose, such as galactose and galactonic acid and galactosamine, were all significantly increased in these failing hearts, as was mannose, as was sorbitol. Now, what is the significance of these changes? It turns out that these metabolic intermediates have been linked to pathophysiology. For example, mannose promotes N-linked glycation, which can lead to remodeling of the extracellular matrix and activation of inflammatory signaling pathways. Sorbitol has been implicated in the depletion of NADPH and glutathione that leads to oxidative stress. And the lactose has really been linked as a profound activator of ROS and age or rage signaling, which can promote inflammation. So this really began to implicate these glucose-derived metabolites in the pathophysiology of heart failure. So just to summarize and what we learned from these experiments was that when we looked at in these failing hearts, we saw that there was accumulation of various glycolytic intermediates, a reduction in TCA cycle intermediates in the heart. And post-LVAD, of course, we saw that this was exacerbated. So we wanted to genetically test this in a mouse model. And so this is work done by Yuan Zhang. When she was a postdoc, she is now a junior faculty with help from Eric Weatherford, to really see, could we model this mismatch between glycolysis and mitochondrial metabolism in the mouse? And it turned out that we had a nice target that we could use to address that, which was the mitochondrial pyruvate carrier that had been recently cloned at the time. And mitochondrial pyruvate carrier mediates the uptake of pyruvate into mitochondria for further metabolism in the TCA cycle. So what we generated were mice that were deficient for the mitochondrial pyruvate carrier, and so the prediction would be that there would be accumulation of glycolytic intermediates, pyruvate, lactate, and a reduction in TCA cycle intermediates. So here's the mouse. Mitochondrial pyruvate carrier exists in a complex. There are two genes, MPC1 and MPC2. We knocked out MPC1, but MPC2 is degraded because they live in a complex. So loss of MPC1 leads to loss of both subunits of the mitochondrial pyruvate carrier. And so you can see that there's a profound reduction in mitochondrial pyruvate uptake, as shown here. And also when we look at mitochondrial oxygen consumption using pyruvate as a substrate, there's a significant reduction in oxygen consumption rates when mitochondria are exposed to pyruvate. However, oxygen consumption rates for other metabolic substrates were unchanged. And in fact, we saw that there was a redirection of glucose-derived metabolites. So this is a Western blot looking at O-GlUCNAC-modified proteins. This is quantified here. You can see that there's a significant increase in O-GlUCNAC modifications, as well as an accumulation of glycogen in these hearts that was seen most strikingly in the fed state. So what happened to these hearts? Well, these animals are born normal. Their hearts are normal at weaning, but by 8 weeks of age, they get pathological left ventricular hypertrophy with preserved ejection fraction, which transitions to a dilated cardiomyopathy and heart failure as these animals get older. And so you can see here, looking at the ejection fraction by echocardiography, that as these animals transition from 8 weeks to 18 weeks, there's a significant reduction in ejection fraction, and then they have increased mortality, more than likely from heart failure, as they get into their middle age. So we have a model now where we have impaired the ability of the hearts to oxidize glucose. And so we said, well, were they failing simply because they just didn't have enough glucose to eat? What if we gave them an alternative substrate? And so Yuan designed a series of experiments where she decided to put these animals on a ketogenic diet, which in the mouse is a low protein, low carbohydrate, high fat diet. And we treated animals at weaning to see if this would prevent the heart failure, at 10 weeks to see if it would reverse the pathological cardiac hypertrophy, or at 18 weeks to see if it would reverse the heart failure, and then at 24 weeks. And so in up to the age of 18 to 20 weeks, we were able to prevent heart failure or completely reverse hypertrophy or reverse heart failure when the animals are put on a ketogenic diet. As the animals aged and got cardiac fibrosis, we still saw a partial but not a complete rescue. So let me show you one example of these data. So these are animals that were started off at the time when they had heart failure. You can see that the knockout animals in red had reduced ejection fraction, but within 3 weeks of being on the ketogenic diet, and certainly by 8 weeks of being on the ketogenic diet, they completely normalized their cardiac function, they normalized their LV mass, and this is the heart rate normalized the tibial length. You can see the cardiac hypertrophy which they got while they were on the normal chow is completely reversed on the ketogenic diet. This rescue remains as long as they stay on the ketogenic diet, because when we took the animals off the diet and switched them back to normal chow, their hearts failed again. So here's how that study was set up. So animals were treated on a ketogenic diet for 10 weeks, so their hearts were rescued. We then randomized them to either remain on the ketogenic diet or to go back to normal chow. And so you can see that the animals who were switched back to normal chow again developed cardiac hypertrophy, whereas the animals that remained on the ketogenic diet did not. They developed heart failure with a reducing ejection fraction, whereas the animals on the ketogenic diet did not. And again, you can see the heart rate to tibial length increase in cardiac weight, which occurred when the animals were switched back from the ketogenic diet to the normal chow diet. Now, this is a complicated slide, but I will walk you through it. And so we really wanted to look at what was happening metabolically at a deeper level. And so we did metabolomics in these mice in response to a ketogenic diet. So we have 4 groups here. We have the controls on normal chow, the knockouts on normal chow, the controls on a ketogenic diet and the knockouts on a ketogenic diet. And so this is looking at acyl carnitines. And one of the things that you would see immediately is that on a normal chow diet, looking at the blue bars here, the knockout animals had accumulation of acyl carnitines. And so we interpreted this to mean that maybe they were increasing their dependence on fat metabolism, although the fat metabolism was incomplete. When we put control animals on a ketogenic diet, shown here in green, we also saw an accumulation of acyl carnitines. And of course, this big accumulation of C18 is because the predominant fatty acid in this diet is oleate. And so you can see that the wild-type animals are also now accumulating acyl carnitines to the same extent as the MPC mutants on normal chow. But note that the mutant animals on the high-fat diet did not increase their acyl carnitines any further, suggesting that they already had the capacity to increase the oxidation of fatty acids. Importantly, we also saw that there was a very dramatic switch in glucose utilization. So focus here on free glucose levels. You recall that when we knocked out MPC1 and had these animals on normal chow, there's an increase in free glucose, which is not unexpected because there's this backup in glycolysis. However, on the ketogenic diet, this was in fact abrogated. And perhaps it's seen most dramatically here that the accumulation of pyruvate and lactate that occurs in these MPC animals on normal chow was completely prevented on the ketogenic diet. So said another way, these animals completely shut off glycolysis and their hearts were essentially living on ketones and fat. But this actually, led to a rescue in energetics. You can see here that the reduction in ADP and ATP levels in the hearts of MPC mutants on normal chow was completely normalized when they went on the ketogenic diet, as shown here. And these changes also correlated with a reversal in the accumulation of these glucose bypass pathways. So, for example, shown here is O-glucnac, which accumulated in the MPC hearts on normal chow, which was reversed on the ketogenic diet. And similarly, the accumulation of glycogen, which you can see here in the MPC animals on normal chow, were completely normalized when these animals were placed on the ketogenic diet. So we did one other experiment in these animals. We subjected them to an additional stress, which is pressure overload. And what you are seeing here is an experiment where we take either control mice or MPC mutant mice, and we subject them to pressure overload by transverse aortic constriction. And so you can see that the knockout animals shown here have much more fibrosis. So we said, okay, so these animals clearly have less reserve to respond to a hemodynamic stress. And therefore we said, well, could the ketogenic diet rescue that? And so in this experiment, we put animals on a ketogenic diet at the same time that we induced pressure overload. And in fact, that had no effect on survival. So you can see here in the red that the animals on the normal chow, the knockouts on the normal chow, had reduced survival, premature death. But animals that were placed immediately on a ketogenic diet at the time of pressure overload also did not see any improvement, suggesting that it wasn't just simply giving them an alternative metabolite that was responsible for the rescue, which I described earlier. So what Yuan then did was actually take another group of animals and put them on a ketogenic diet for 3 weeks before pressure overload. And what in fact, so here's what we see in animals in normal chow, is that when we do pressure overload, again, we see a dramatic reduction in ejection fraction in the MPC animals on normal chow diets. But when the animals were fed a ketogenic diet for 3 weeks prior to transverse aortic constriction, in fact, they maintained their cardiac function after transverse aortic constriction-induced pressure overload. So what this therefore tells us is that left ventricular remodeling is not just solely dependent on the availability of the alternative substrates, but really describes a broader remodeling of glycolytic metabolic pathways that promote cardioprotection. And very recently, myself and colleagues at Utah also show that this is true in human hearts. And so, again, these are humans, and essentially what was done was, again, the individuals received an injection of a heavy label of glucose prior to insertion of a left ventricular assisted device. And in fact, and then there were some patients who clearly showed recovery, and so they had another experiment done to look at these flux pathways. And this, in fact, what we saw was that in the hearts that recover, there is a remodeling of glycolytic pathways leading to a reduction in sorbitol, an increase in 1-carbon metabolism that may promote more efficient synthesis of biomolecules, and increase in pentose phosphate pathway, which we believe really is promoting cardioprotection and repair. And this occurs in concert with a reduction in oxidative stress, which we think may be a metabolic marker of myocardial recovery. So what I try to kind of convey in this part of the talk is that we think that there is important role for metabolites from glycolysis that certainly play an important mechanistic role in driving ventricular remodeling in heart failure and potentially in driving myocardial recovery when as shown here. And so we think it's actually really quite exciting because number one, it might also explain the increased risk of heart failure and diabetes, but I think now it's beginning to identify potential pathways that we could potentially attempt to modulate to increase to improve the outcomes of individuals with heart failure. So with that, I want to end, and I want to acknowledge members in my lab. Normally, we have a nice group picture every year, but because of the pandemic, we had to kind of socially distance. So everybody submitted a selfie this year. So this is Yuan's selfie. So Yuan again really directed and drove the studies in the MPC project, and we have important collaborators who have contributed to this work. Gene Schaefer from WashU, who shared with us the acyl-CoA synthase mouse. Eric Taylor, who was one of the original investigators who cloned the mitochondrial pyruvate carrier. Stavros Drakas from the University of Utah in the studies of the human LVAD patients, and Ken Margulies from the University of Pennsylvania, who very generously shared with us the heart failure samples that I showed you the metabolic profiling on. So with this, I will end my talk. And again, thank you very much for the opportunity to share some of our data with you and to celebrate your 75th anniversary. I'll be happy to take any questions. Thanks.
Thank you very much, Dr. Dale, for this great talk, full of exciting results. It was really nice. So first, I would like to invite the audience to make some questions using the microphone or the video. And then also I can read a few questions from the chat.
Okay.
Okay.
So, hello, Dr. Dale. Thank you for your amazing talk. Really exciting indeed. I was just curious. You showed that the MPC deficiency had an age-dependent reduction in the ejection fraction.
Mm-hmm.
Correct?
That is right.
Yeah, and I was just wondering, what prevents MPC deficient mice from developing heart failure early on?
Yeah, that's a very good question. So, and I will refer to our recent publication. So it turns out that early on, within the first 8 weeks of life, there's actually a very striking adaptation where there is MPC independent pathway that gets glucose-derived carbons into the TCA. We aren't sure exactly what that is, whether it's going through alanine or through malic enzyme. But for a short period of time, they have this adaptation that gets these carbons in. It does come at a cost. It comes at a cost of energy. It comes at a cost of anaplerosis, but it's not sustainable. So at some point, this then begins to fail, and then that's when we actually see this transition from kind of compensated hypertrophy to heart failure. So that's the reason why it doesn't occur immediately. There is an adaptation that attempts to bypass this defect, which for a short period of time maintains LV function in these hearts.
Okay, amazing. Thank you.
You're welcome.
Good. More questions, please?
Hi, I have a question.
Yes.
Can you hear me?
I can. Yes, Carissa,
Yeah. So it was very, very interesting. Thank you for all sharing. And I was very curious about the OPA1 and in white adipose tissue. Is it possible that in the condition where you have the diminishing of OPA1 in BAT, can you start the expression of OPA1 in white adipose tissue?
Right. So, I think that's a very good question. So, just to kind of again clarify what we did. So, we used the uncoupling protein 1 promoter to reduce OPA1 in brown fat, and it turns out that that promoter expressed in brown fat, but it doesn't really express very much in either white fat or even beige fat, right? And it turns out that in the white fat of those knockout mice that underwent browning, we did actually see an increase in OPA1. So again, I think OPA1 is another marker of kind of increased mitochondrial fitness and increased mitochondrial biogenesis. And so we in fact saw that OPA1 was being increased in the white adipose tissue of the brown fat deficient OPA1 knockout mice.
Okay, that's interesting. Thank you.
Great. More questions?
I would just complement the Clarissa question, please.
Yes.
Again, with this OPA1 knockout, it's really amazing because how you have like kind of whitening of the bat and browning of the what, let's say, and they still are able to maintain their core temperature and are resistant to gain weight induced by diet. And what does it tell us about the role of fat and the role of white or of the beige tissue to those metabolic adaptations, energy temperature, and it's amazing.
So I think it tells us a lot. First of all, it tells us about how organisms are wired to promote homeostasis. That's the first thing. Secondly, I think it also identifies really the important endocrine role of brown adipose tissue beyond just its classical thermogenic role, because clearly what we learned from this model was that as brown fat was becoming impaired, it had an endocrine response that then promoted a homeostatic adaptation in white fat that almost substituted for the lack of effect or for the impairment of the brown adipose tissue. So it also taught us about kind of inter-tissue crosstalk on the one hand.
Yeah.
And I think that it also told us that there, and I didn't show you all the data, but we have done RNA sequencing, and there are a lot of other secreted molecules coming from brown fat that we're now trying to figure out what the metabolic consequences are. So really, I think the message is that brown fat is very important in 1 second. Let me just stop this. There we go. Yeah, brown fat is very important in maintaining metabolic homeostasis, not just simply from its endogenous function in terms of thermogenesis, but also an endocrine function, as I think we showed here.
Very interesting. Thank you.
Okay. More questions?
I think there's some questions in the chat.
Yes.
Okay.
I can read or for you, or you can you read that? Yeah?
Well, let me see. Let me see if I can here.
I can read for you.
I can see. So let's see. Somebody asked, Marcelo Lamas asked, concerning the increased content of diacylglycerol in the heart, do you know if there's a higher activity of DAG kinase which would contribute to increased phosphatic acid and lysophosphoric acid? Is there an involvement of lysophosphoric acid in these pathways? So I don't know the answer of that question, Marcelo, because we didn't directly measure the activity of DAG kinase. What I can tell you, though, is that when we did lipidomics on the mitochondria of these animals, we saw that there was significant remodeling of the side chains of cardiolipin. But specifically to your question, we did not measure that kind of activity. It looks like, and then Isis Trebenzoli had a question, and she said, thanks for the talk. Thank you very much for your kind words. Are the OPA1 knockout muscle or bat resistant to cardiovascular complications on the high-fat diet too? And how does the FGF21 signaling look like in the heart? So it's a very good question. So we're actually now doing those experiments. So I don't have an answer for you as yet, but we actually are looking now at both vascular as well as cardiac implications in the context of high-fat feeding. So stay tuned. Maybe the next time Carmen invites me down, I can tell you that story.
Yes. Yes. Okay. More questions? Let's see. Okay.
So there's one last question from YouTube, from our viewer on YouTube.
Excellent.
So Juliana Camacho Pereira asks, she would like to know if the increase in energy expenditure in OPA1 deficiency mice is related to the availability of mitochondrial surfaces to substrates.
Yes, so if I understand the question correctly, so the question is, these animals clearly, whether OPA1 is deficient in muscle or brown fat, show increased energy expenditure. I think the person is asking whether or not that really depends on the mitochondrial reserve in these other tissues. And the answer is yes. We have knocked out and didn't talk about this with OPA1 in brown and white adipose tissue together, and we don't see these adaptations. So I do think that intact mitochondrial cristae and their function is in fact required for this increased thermogenesis that we are seeing in these models. I see another question in the chat. The increased uptake of ceramide and DAG in the ACS-stringent animals have implications in signaling cascades involving mitochondria. I would say that the answer to that is yes. Others, not us, have also looked at inhibiting ceramide synthesis in different tissues, and I believe that their work has implicated that there certainly are effects of ceramide on both the composition of mitochondrial inner membrane lipids as well as mitochondrial cristae structure and function as well.
Excellent. I think Dr. Bruno Diaz, our director, has a question also.
Yes.
Thank you, Dr. Abel. It was a very nice talk. In several of your figures, reactive oxygen species appeared prominently as part of the mechanism. What do you think of using reactive oxygen species as therapeutic targets? And because it has been tried before several times with no success so far. But do you think there is any hope that it can be used? And why these previous strategies did not produce, relevant results or actually beneficial results?
Yeah, that's an excellent question. Excellent question. And so clearly, ROS is a two-edged sword, right? You do need ROS to activate critical signaling pathways. It's kind of like nitric oxide, right? That too much NO is bad, whereas a small amount is good. And I will refer you to a paper that we just published in October in Cell Metabolism, where we described a phenomenon where basically electromagnetic fields actually can promote a small amount of ROS production in the liver that then leads to a adaptive hormetic response that is really acting like an antioxidant, but at a level that's probably much more finely tuned than you would get with a pharmacologic agent. So I think that part of the failure of pharmacological agents is that it really it's a blunt tool. And so if you end out invariably scavenging what I'm going to call good ROS, then I think you lose many of the potential benefits. So I would definitely encourage you to actually read that paper, because we're actually very excited that we've come up with a way now to tune at a very small degree ROS biology to then lead to a sustained antioxidative response. And in fact, in that paper, we show that we could completely both prevent and treat diabetes with that approach.
All right. Thank you very much.
You're welcome.
Good. So I'm just checking if we have more questions on YouTube also. Just a few seconds.
No, not yet. No. So
no. Okay. Very good. So last chance for questions. No more questions? Okay. So, yes, I'd like to thank you again, Dr. Dale. It was a pleasure to have you here, and thank you. It was, I think it was for us, it's a real honor to have you here in our celebration.
Well, thank you. And I see that Karen just sent a nice note too. Thank you, Karen. And also I should have mentioned in my talk that Karen certainly also had a major contribution in the Muscle OPA1 story when she spent a year with us when we were back in Salt Lake City. So again, thank you, Karen. And hello, Karen, too. Good to see you.
Yes. So thank you all for the audience. Yes. And yeah, I open for your last comments.
Well, again, it's been really a lot of fun to visit with you this morning. As I said, I'm grateful for these platforms that allow us at least to communicate across the oceans. And so, I feel I've been with you a little bit this morning, if not in body, but in spirit. And again, I look forward to keeping in touch with many of you and hopefully seeing you again in person at some point in the near future.
Yes, sure. Thank you.
Thank you. Okay. Ciao.
Ciao. Thank you.
Bye. Bye.
Bye. So I have also a few messages for the students in Portuguese. Então, só lembrando que na próxima semana, na terça-feira, teremos uma conferência extra, colocamos ali também no bate-papo. Então, será terça-feira do professor Farinas, sobre microscopia, às 5 horas, e na quarta-feira, ao meio-dia, será a última conferência do Programa de Fisiologia. Então, agradeço a todos e a todas novamente, até terça-feira.